Welcome to the second part of the WebIDQ video tutorial series. In this section, we'll go over the LIMS module and how to create and register projects in WebIDQ. LIMS is the Laboratory Information Management System module for WebIDQ. It's the starting point for each assay and responsible for registering samples, creating projects and submissions, and finally arranging samples into work lists for LC-MS analysis. LIMS is broken up into three main sections: projects for designing your experiments, samples for registering the samples that will become part of the projects, and finally custom QCs, where you can register your own quality control samples and define expected target values for each metabolite. We'll start with the samples section. Samples are unique database entries that you plan to analyze on a kit plate. These include kit-specific samples like calibration standards and quality controls, as well as your own experimental study samples. While the kit-specific samples are already registered in the database, before creating a work list, the study samples to be included in the plate layout must first be registered into the database. Samples can either be registered individually, which is suitable for small projects or adding an extra sample, or batch imported, which is useful for full kits or larger studies. In the top row of menu icons, new samples can be added, existing samples edited or deleted, or multiple samples can be imported in a batch. The simplest way to add a new sample is to click the plus icon, enter the required sample information in the pop-up window, and save. As mentioned earlier, some of the fields can be searched by typing for quicker entry. You'll see that the new sample is added to the database and assigned a unique barcode number. These assignments ensure that no two samples in your database will ever have overlapping identifiers. You can further define information about your samples in LIMS. For example, clinical information like disease state or numerical information such as patient age or treatment details can be defined as additional metadata. Later, these elements will be displayed together with the final results. To add a new category, click the Add Metadata at the bottom of the sample selection. Click the plus button to add a new attribute. Give the attribute a name and select the type. Categories are used for attributes that consist of discrete groups. For example, control versus disease or distinct time points. Date, number, and text can be used for all other metadata. Categories will need to be further defined by adding the individual group components. Select the category you want to edit, then click the plus in the side pop-up window to add a new group. You can keep adding as many groups as required for your experiment. With a single sample or multiple samples selected in the sample list, use the drop-down menu to assign a group to the selected samples. If you are working with samples that need to be extracted prior to analysis, such as tissue, feces, or cell cultures, a tissue factor or cell number can be defined for each sample. Once these values are defined, they allow metabolite concentrations to be automatically normalized to the amount of starting material before exporting results. Select a single sample or multiple samples and click the tissue and cell factors at the bottom of the screen. The tissue factor is used for samples that are extracted from a starting mass. In the tissue factor field, input the ratio of extraction solvent to sample mass in microliters per milligram. If working with cell samples, put in the cell number in the cell number field and the volume of extraction solvent used to lyse the cell in the cell extraction volume field. Later, when exporting the results, the sample concentrations can be normalized to tissue factor or cell number as defined here. While creating one sample at a time is fine for small projects, if you have a full plate or a larger study, you'll want to do a batch import of your samples. To import a batch of samples into the WebIDQ database, use the Import Sample button. In the pop-up window, drag a CSV or text formatted sample list into the upload box. At a minimum, only a sample ID column must be present. Other metadata such as species or material can be defined during import. If your samples require tissue or cell extraction information, these can also be included as separate columns in the CSV. If you choose to define the material or species groups in the CSV, you must make sure that the syntax matches those in the database. For a full list of possible names, you can check the drop-down list from each category. Once the file is imported, assign all the columns from the CSV to categories that are present in the database. If no material or species were defined in the document, they can be assigned globally for the whole import. WebIDQ will try to guess which columns correspond to which categories from the database. If it guesses incorrectly, you can deselect these by pushing the X button. Other database categories or custom groups and variables can also be assigned here, but are not required. Furthermore, not all columns from the imported file need to be assigned and can just be left blank. WebIDQ should automatically determine the correct format, otherwise the delimiter can manually be adjusted in the import window. Before continuing on, you must also assign a contact for the entire import. You can think of the contact as the owner of the samples. You can choose a contact from the drop-down menu. Otherwise, you are also able to create a new contact by clicking the plus icon. Click the Next button. In the subsequent window, confirm the sample list is OK and store. Now that we have all the samples registered, we can move on to creating a project. The project section is where the previously registered samples can be organized into plate layouts and finally exported for the MS instrument. In the project section, you can click New Project to start a new project. Otherwise, if another project has already been selected, you can just click the plus symbol. In the pop-up box, provide the new project with a unique code and project name. Optionally, you can also enter a description or assign a contact to this particular project. If no contacts are already in the database, a new one can be added here. You can also optionally assign which WebIDQ users have access to the current project by assigning them in the Project Teams field. If you leave it empty, all users will be able to see and edit the current project, while admin accounts can always access everything regardless of the project team. Once everything is entered, click Save and the new project will appear in the left sidebar. With the new project selected in the left sidebar, start a new submission by clicking the New Submission button. In the pop-up box, give the submission a name and optionally add a description. You should be able to see that the new submission is created as a folder within the selected project. As your projects progress, you may need to add additional submissions. You can add a new submission by clicking on the project again and then clicking the New Submission button. New submissions will be added as folders as part of the same project. In general, submissions can be thought of like individual plates. For best organization, it's usually easiest to keep one plate per submission. Work lists, which are contained inside submissions, can be thought of as the actual plate layout of the assay. The work list determines which samples are included in the analysis and how they are arranged on the plate. Once you've created a work list, it can be exported to the LCMS system as a sequence or batch file. With the submission selected in the left sidebar, click on Generate Work List. In the pop-up box, select the OP for the assay that will be prepared according to the respective kit user manual. OPs, or operating procedures, contain the specific processing instructions for the assay that you'll be running. You must link at least one OP to the work list that you're preparing. Each type of kit will have its own unique OP code depending on the combination of assay and instrument being used. For profiling kits, such as the Quant500 XL, which requires multiple OPs, you can select all OPs at the same time and continue. In the next window, you need to select which samples will be included with the assay. Samples are organized by type in the top menu bar. Follow the plate layout according to the respective kit user manual that you're preparing. Samples can be sorted by clicking on the table headers or searching using the filters next to each header. Continue on to link the zero sample, calibration standards, QCs, and unknown samples in order. Multiple samples can be selected and dragged to the plate at the same time. Alternatively, you can click the Add Selected Sample button to add the selected samples to the bottom of the list. To remove samples, simply drag them from the selected samples back into the top part of the window. For samples that must be run multiple times, such as the zero sample, you need to add it for each time that it will be injected. For samples such as the QC Level 2, which is recommended to be run in replicates but must be distributed across the plate, another option exists. Select the QC Level 2 from the list. In the pop-up side menu, increase the number of distributed replicates. In the final work list, the selected sample will be replicated at even intervals across the plate. If required, the amount of injections can also be changed for the selected samples in this side pop-up dialog. In the pipetting mode drop-down menu, sample pipetting orientation can be changed between horizontal and vertical depending on your preference. This choice will determine if the plate is pipetted from top to bottom or from left to right. Keep in mind that for starter kits and half kits, the orientation must be set to vertical. Finally, if you wish to randomize the samples, this can also be changed at this time. Only study samples will be randomized. Biocrates samples like QCs and calibrators will stay in the same order. When finished editing the worklist layout, click OK to proceed. Once the worklist finishes being generated, it can be selected underneath the submission in the left-hand sidebar. You can select the individual plate runs to edit the worklist further. You can mouse over the samples to show some additional information. You can right-click on the samples to copy, cut, delete, or select all. Right-clicking on an empty well allows you to insert an additional sample or paste a copied or cut sample. Selecting a sample or multiple samples from the bottom sample list can be used to update the sample volume that will be added to the plate or change the number of injection replicates. You can also drag the samples around to rearrange them. When dragging one sample onto another sample, they are swapped. When dragging a sample in between wells, the sample will be moved in between those two positions. If required, additional plate runs can be created by right-clicking on the available plate runs and copying the plate run. For profiling kits with LC and FIA components, the calibration standards can be optionally removed from the FIA plate runs since they are not used in the quantification process and are not required to be collected. Select all the calibration standards, then right-click and delete to remove. Once all the changes have been made, select Export Worklist to MS. In the pop-up window, select the MS type and auto-sampler according to your instrument configuration. You can choose to randomize the sample injections. Again, this will only affect study samples, and biocrates samples like QCs and calibrators will be injected in the same order. Depending on your auto-sampler configuration, you can also change the rack position for each of the plates that will be run. For example, in the Quant 500 XL kit, where the FIA and XL plates are run back-to-back, you can set the FIA to position 1 and the XL to position 2 so that both can be injected at the same time. Finally, if you would like a plate layout PDF, select the option to print the plate report. Now you can export to download to your local computer. A zip file will automatically be downloaded. Extract the contents onto the LCMS operating PC. A text or CSV file will have been generated for each kit plate worklist. These can be imported into the MS software before starting the LCMS run. The download will also contain another folder containing a PDF of the plate runs. This can be printed out and taken into lab as a template while you're preparing the kit. This concludes the second part of the WebIDQ video tutorial series. In this section, we looked at how to register samples and make a project in LIMS before exporting it to the MS instrument. For additional details and information, please consult the WebIDQ user manual. In the next section, we'll look at how to import and quantify the raw data files in results.